Hello, everybody. Thank you and welcome for attending our webinar on SomaticSeq, a machine learning approach to accurately detect somatic mutations. My name is Anoop Graywal. I'm Product Marketing Manager here at Bina Technologies, which is part of Roche, and I'll be co-presenting with our Senior Scientist of Bioinformatics, Li-Tai Fang, who's a primary author of this algorithm, machine learning-based algorithm, SomaticSeq. Just a reminder, we'll follow our oral presentation with a WebEx question and answer session, and so you should see a horizontal green bar at the top of your screen. When you're ready to ask a question, just hover over that, and that'll bring down a larger gray bar. Click Q&A from there, and that'll open up a chat box specifically for questions and answers. Enter your question in the Q&A chat box, and then with respect to recipients, choose to ask all panelists. That way, multiple of us here will see that and be able to respond to your question. If you happen to be on a mobile device, your interface will look slightly differently. Just select Q&A from the top right menu. Your Ask a Question box will open up. Enter your question. Click Send from the upper right. Again, make sure that you have all panelists selected when you're asking the question. And after you've sent the question, you'll be able to toggle between My Q&A and All Q&A so that you can see questions that others are asking. All right, let's get started. Now, somatic mutations are those mutations that occur other than those that occur in the germline, so within the organism during the course of their life. And particularly, we're interested in detecting somatic mutations that arise due to cancer. Some of the challenges with respect to detecting those mutations is that there is tumor heterogeneity. So some cancer cells, but not others within a tumor, may carry a mutation. Somatic mutations are also rare on a genomic scale. When you look at germline variants, they occur at a rate of about 1 in 1,000 with respect to SNVs and small indels. But somatic mutations are much rarer and therefore harder to detect in a larger pool of potentially, for instance, false positives. The other issue one's dealing with is that ideally you would like to isolate the tumor tissue away from the unaffected tissue and detect the mutation, but you're often, that sample or biopsy is often contaminated with control tissue, and therefore you have even lower representation of the somatic mutation. So here's a screenshot of a potential tumor normal sample in IGV. And again, when you're calling somatic variants, germline variant calling is already challenging. Somatic variants is especially challenging because you can't assume a allele frequency distribution of 0%, 50%, or 100%. If your tumor tissue is contaminated with control, that's going to throw off the representation. And remember, again, within the tumor there will be heterogeneity. Some of the cells may carry the mutation and some not. And then in a lot of these samples arrive as formalin fixed. The biopsy may have been collected some time ago, and you may not have a lot of tissue to work with. So we have issues of DNA quality and quantity to deal with. That's one of the reasons why the DREAM Challenges put out a competition or a challenge to the research community to come up with better ways of detecting somatic mutations. The DREAM Challenge is a non-profit collaborative community organized by groups such as SAGE, Bionetworks, and IBM. And with respect to the Somatic DREAM Challenge, they were joined by ICGC, the International Cancer Genome Consortium, and TCGA, the Cancer Genome Atlas. And so Bina Technologies participated in the Somatic DREAM Challenge. Specifically, we participated in the challenge to detect somatic indels and somatic SNVs. The results were, for stage 5 of that challenge, were released recently, and among a number of well-known groups, Bina scored first with regard to somatic indel detection. The score you see here is an F1 score. It's a harmonic mean of the precision and sensitivity, so it's a good balanced measure of accuracy. And we came in second with respect to SNV. You can see with the indel score, we achieved a harmonic mean or F1 score of 71.6%. Indels are just by their nature harder to call. And with respect to somatic SNV, we had a respectable 99.7%. Now, Lee Tai Feng, my colleague, will get into the details of how we were able to do so well. Lee Tai?
Thank you. My name is Lee Tai. I will talk about the SomaticSeq workflow that we have developed here at Bina Technologies. The source code for the program is freely available under BSD open source license. And so you're free after the webinar to download and try it out yourself. The methodology and the results of the validation are also published in Genome Biology, where we have described the results and the rationale behind the workflow. So feel free to test and look for yourself. So I'll talk about Dream Challenge and how the data is generated. So Dream Challenge, basically, they would take a band file with realignments of a high coverage, let's say 60x, and then randomly split the reads into two smaller files, two groups of files. One is designated the normal, and the other one is designated the tumor. And so in these two files, they came from the same sample, came from the same sequencing experiments. In theory, they should have the exact same genotype. Now, in the designated tumor, some synthetic mutations are spiked in at different fractions. So in the end, you know what are the true somatic mutations where you can evaluate your methods based on the synthetic mutations. Now, in early days of next generation sequencing, we have genotype callers that was developed before somatic callers, and I and many other people have tried to look for somatic mutations by a simple approach, now termed naive subtract. Basically, we would run the genotype, the germline genotypers on the tumor and the normal, and then try to see the difference in the results. Now, the problem with that is, as Anoop has described earlier, that in tumor, you have a lot of heterogeneity. Some mutation occur at a very small fraction, and so germline genotypers will not detect those. So you have a lot of false negatives. Another issue is that germline variants outnumber somatic mutations by orders of magnitude. So on this figure here is a little experiment we did with the Dream Challenge data. In this experiment, we have basically masked all the somatic mutations. So the only thing you have in those two files, one is tumor, one is normal, are really should be identical. So if your caller is perfect, then they should have identical genotyping results for each one of them. The result is that they are, in fact, very close. So when we run haplotype caller on the two files, they have over 95% concordance. But you do have about 160,000 that occur in only one file, but not the others. And all of those really are germline variant false positives if you're looking for somatic mutations. And the reason is simply that germline SNPs outnumber somatic mutation by orders of magnitude. And so even a low level of false positives would overwhelm your true signals. And in the past few years, there have been development in many somatic mutation callers. Now, those are more sophisticated than try to look for somatic mutation with naive subtract. Some of the things, some of the tools we have incorporated in our own somatic workflow, for instance, Mutec, VARScan, joint SNV mix, Somatic Sniper, Vardig, and then there are a lot more. Each of them will compare the tumor and the normal data simultaneously to determine if a variant is a somatic mutation. This is a Venn diagram for each of those 5 tools, also run on Green Challenge. If you run multiple tools, you can increase your accuracy and sensitivity. In this data set, there are about 8,000 true mutations. We know they're true mutations because they're synthetically spiked in, and you can increase your precision by taking intersections, by taking a consensus call. For instance, in the middle, you have about 5,000 calls that's agreed upon by every single tool, and you can increase your precision by taking a consensus. A problem with that is that based on the nature of the data, based on the characteristics of the sequencing data, what is the best combination is not always the same. So for one data set, you may find out that Mutec and VARScan gives very good results, but on a different data set, you may find that, joint SNV and Vardig, in fact, give you better results. And so that's why we have developed SomaticSeq, and the detailed results for consensus evaluation is also in the paper that we have published. The workflow for SomaticSeq can using the band file and multiple tools. So the first part of the analysis, we use the so-called Broad Institute's Chdk best practices. We align the FASTQ files, the reads, with BWA-man. We mark the duplicates. We do in-depth realignment and base quality recalibration, and then we get the band files for the tumor and the normal. And then we run each of the 5 tools on the band files, and we get a combined call set from all of those tools. Now, for each of those calls, we look back into the band file and get features related to its mutation status, such as strand bias, mapping quality, base quality. And then we use a machine learning algorithm to train, if we have a ground truth, to find, really, to build a classifier of what is a true mutation, what is false positive, based on each of those features. And then that classifier can be used to generate a high confidence call when you have your own data to test. So I'll talk about the machine learning algorithm and the features that we have used. So the machine learning is based on over 70 features. Some of the top features include caller classifications. So that's simply a list of calls, say, Mutec called it, VARScan called it, Vardig did not, and et cetera. So caller classification, and we also have things like mapping quality, strand bias, p-value, facial exact test score that's reported by Vardig, forward, reverse, read count supporting variant and reference calls. So in that information, there is the quantity of evidence. So the more read counts you have, the more evidence you have, but you can also infer strand bias from this information and prior probability, prior knowledge such as DB-SNP membership. A call in DB-SNP is more likely to be a germline positive. So we can briefly kind of look at 4 of the features and see how they can help you, to determine whether a call is true or not. On top left is a histogram generated from Dream Challenge. You can see that the false positive outnumber true positive significantly, but they have different distributions. So the number of mismatches of the read to the reference. So if you have a perfect read that is all reference calls, you will have a mismatch of 0. And if you have a perfect variant read, for instance, you're going to have a mismatch of 1, and that mismatch is the variant call. All other sites are reference. And if you have more than 1 mismatch, then you're essentially likely to be a misaligned read somehow. So, so for mismatch of 1, you can see that the false positive to true positive ratio is about 3 to 1. You still have more false positives, but you have a lot of true positives. Now, if you look at the number of mismatch, number of mismatches, that is 2. Now the ratio goes from 3 to 1 to something, by eyeballing, it's like 10 to 1. And when it's a mismatch goes to 3 to 4 and 5, then essentially there is no true positive. Everything here is a false positive. Basically, it's saying that if you have a variant call, but all the reads have more than one mismatch, you are likely to see a read that's misaligned instead of a true mutation. So that is a strong indicator using a feature. On top right, that's mean base quality score for the variant site. So essentially it's saying that if your base quality score is very low, below 30, below 32, you're more likely to see false positive than true mutations. Lower left is another metric. This is the Z-score of base quality rank sum. So basically that is the statistical test for the base qualities of variant reads against the base qualities of reference reads in the same position. If the two group of reads have the same base quality score, either they're both very high or both very low, you will have a Z-score of 0. If you have variant reads that's of lower quality than the reference reads, this is really saying maybe they're not variant reads, they're, the bases are not called correctly, then you will have Z-score in the negatives. So from this histogram, you do see that all the true positives have, essentially a bell curve centered right around 0, and the false positives have a huge peak around negative, indicating that you, in addition to having high base quality, you want to have base qualities that are more or less consistent between your two reads. And dbSNP membership is also an indicator for germline false positives. You do have actually a few true positives in dbSNP sites, but they are much fewer than compared to non-dbSNP sites. And the machine learning algorithm will use each of those 70 features and generate an ensemble of decision trees, and each decision tree is used to classify a call. So here I'm just showing an example of the highest weighted decision tree in the classifier that is built based on Dream Challenge SNV call sets. So this decision tree has a weight. So it will really go like this. You have a call, and then it's going to ask the mutate call it. And if the mutate did not call it, then you say, well, what's the var scan score associated with this site? And if it's less than 27.5, you say that's a false positive. If it's greater than 27.5, then you ask, is it a dbSNP site? If it is in a dbSNP, then you say it's a false positive, it's probably germline variant instead. If it is not in dbSNP, then you would classify that as a true mutation. And then you have another branch for if mutate call it, but var didn't call it, then it's still probably a false positive. This is one of many decision trees, and there are many trees like this. Each of them has a weight, and you kind of add them on, and to get a final probability score for each of the mutation candidates. And so once you do that, you get a histogram of all the calls, each of them with a probability. So this figure on the left and on the right is generated based on Dream Challenge. So Dream Challenge, I did not mention earlier, has many stages. Each stage is supposedly to get more complex. So for most of the data we talked about here are based on stage 3. But this data set is trained on stage 2 data, and we use the classifier from stage 2 and then applied it to stage 3 and get the results this way. So on the left, you see a histogram of all the calls with different probability scores. And the histogram is color-coded. The blue bars are false, true positives. The red one is false positive. And you can see that just from this graph, they separate very clearly. There is something in the middle between 0 and 1, but vast majority cluster around 0 and 1. And there are false positives around 1. There are also true positives around 0. But by far and large, SomaticSeq has classified those mutations correctly. On the right is a way to see how you change the results if you pick a different probability as a threshold. So the red line here is the recall to sensitivity. The green line here is the precision. So as you demand a higher probability score, then you get higher precision but lower sensitivity as expected. But the harmonic mean almost stays constant through a very large range, saying that SomaticSeq's algorithm is quite robust. It doesn't depend too much on the threshold that you pick. You can pick anywhere essentially between 0.2 and 0.8 and get results that's about the same. Here I'm just showing an example of SomaticSeq's machine learning and how it uses those features to filter out reads. So this is just an example of what appears to be a T to C mutation over here. The reference C, you have some, the reference is T, and you have some reads that's C, and it has good base quality score and apparently pretty good mapping score, and it is called a somatic mutation by each of the 5 tools, but it is actually a false positive, and it is correctly filtered out by somatic score. It has a probability of almost 0. It's 1 to the 10 to the minus 7, about as low as you can get. And the reason is that if you look at the entire read for those variant reads, you can see, now remember that one of the features we talked about earlier is the number of mismatches. If you look at the variant reads, the first one, so besides one here, you see a mismatch here, a mismatch here, a mismatch at G, a mismatch at C. So you have a mismatch. Here you have 5 mismatches here, 3 mismatches on for the other reads. And when you look at the reference reads, they don't have those mismatches. So here you have mismatches for variant reads that's average about 3 or 4, indicating that they're not correctly aligned. Now, if you look at the reference sequence for this position, it is a very low complexity region. You have many repeats, really evidence for that those reads are not aligned correctly rather than real somatic mutations. So we have validated our SomaticSeq results with Green Challenge data, but with different settings. I'll describe what the label on x-axis means. They represent 4 different settings. So setting A is when we take the data directly from Dream Challenge, where normal is pure normal. Tumor has 3 subclones, but it's a pure tumor. Now, in setting B, we make the data a little bit more complex. We spiked in 5% of the tumor into the normal to make the normal more contaminated. So it's a more challenging data. And for setting C, we put 30% of normal into the tumor to make the tumor more contaminated. And the setting D is the most challenging data set we have here. This is when we cross-contaminate normal and tumor to make both samples contaminated. And the green bar on top is the F1 score from cross-validation. And as the data gets more complex, the accuracy decreases, and all the way to D, but it's still a very accurate result, and it is more accurate than any of the individual tools that has incorporated. Similar results for insertion deletion. As you go from easy data to challenging data, the accuracy decreases, but SomaticSeq also still performs quite well. We have, in addition to Dream Challenge, did some in silico titration to have a more realistic data samples. So here we actually have two different whole genome sequencing data, really two different genomes, and designated one of them to be normal and the other one to be tumor. So when they have different genome types between them, it is sort of a virtual somatic mutation, and we also cross-contaminate between each other to generate, a normal that's contaminated with tumor and tumor that's contaminated with normal to have more realistic data types. And we get similar results. I'll describe what's on the x-axis. So N0T50 here means that the normal has variant allele frequency of 0. Tumor has variant allele frequency of 50, and so that is the pure normal and pure tumor, and also there's only one subclone in tumor, and that's a 50% variant allele frequency. So that is the simplest case. So the F1 score for SomaticSeq here is, like nearly 1. Not unexpected because the data is very simple here. The most challenging data set is the last one, N2.5T15. It means that in the normal you have expected variant allele frequency of 2.5% coming from the tumor, where the tumor's variant allele frequency is 15% due to contamination of the normal. And then your F1 score is lower, but still quite good comparing to each of the individual tools. Now, I forgot to mention that the coverage for this data set is about 40. So at 15% on average, you have about 4 or 5 reads of variant reads. When you have coverage below that or due to sampling of different reads, sometimes you do not see a single variant read, so you can't really get 100% sensitivity. Indel is more challenging as you get less allele frequency, you get lower accuracy, but it's still a more accurate than each of the individual tools. We have also validated SomaticSeq on some of the data we have downloaded, real data, and we tested its result against a validated data sets. 2 data sets here. One is Colo 829, that is a melanoma cell line. CLL1 is a leukemia cell line. So for Colo 829, for instance, there are about 450 validated mutations. So we use the classifier that we had built from Dream Challenge, and then use that to call mutations here. And in this instance, we get this many, 37,000 some mutation call sets, and with a sensitivity that is almost as high as any of the sensitivities here. We cannot really get precisions here because we don't have a ground truth of how the true mutations are. We only have a subset of validated mutations. And the sensitivity we get here is also maybe a lower estimate because, when researchers do their validation mutations, they're doing it off a call set, and that call set could be, any of the things that we have incorporated. So you're not going to see validated mutation outside that call set. And CLL1, we are also able to get a sensitivity that is about as high as any of the individual tools, and with the nut size that's only 2,000 versus some other tools that call twice and 10 times as large, implying that SomaticSeq can maintain the sensitivity with very minimal sacrifice in maintain the sensitivity and without really sacrificing the sensitivity while reducing the color size, implying that it has a higher precision. And so,
okay, thanks, Leetai. Now, with respect to the somatic callers, I'll let you know where you can find them in the Bina Rave software. SomaticSeq, as Leetai has mentioned, is available via GitHub, and it will be available in Bina Rave in an upcoming release. But when you are working with tumor normal samples in our solution for secondary analysis, Bina Rave, you will start by generating the BAM files by first running the samples through your FASTQ files through the DNA analysis workflow. If you've got whole genome within DNA analysis, select whole genome analysis, and that will generate the BAMs that you will then specify as input when you subsequently run the tumor normal calling workflow. That interface is quite similar across the different workflows and relatively easy to understand. Specify some metadata. The list of resources are provided, or select some of your own reference files. And then further down the screen in the tumor normal calling workflow, you'll see the options for somatic calling. But just before that, let me just go to the DNA inputs. You'll specify your DNA inputs, and here's where you'll specify the BAM file for each of your tumor and normal. All right. You can see here we have the 5 somatic calling tools that were mentioned as part of machine learning algorithm, as well as somatic indel sector. So you have a choice of a total of 6 tools. By default, in the current version of Rave, version 2.7, 4 of the 6 are selected. You can go ahead and select all 6. It's just that the other 2 are a little bit more computationally intensive, but there's no harm in running those as well. As Leetai already mentioned, SomaticSeq is available in GitHub under the BSD open source license, and a lot of the results he presented are also found within our SomaticSeq publication available via open access in genome biology. You can find more results if you want to drill down even further to take a look at, to understand the performance of SomaticSeq even more. Now, I just wanted to mention that if you would like to try Bina Rave or our solution for tertiary analysis, Bina AIM, and you're an academic or working for a nonprofit, consider applying for a Bina research grant. If you have a next-generation sequencing project in mind, the grant is supplied with respect to a given project and is available for the duration of your project. You can have a whole genome sequencing, whole exome sequencing, RNA-seq, or targeted panel-based project. We say here about 6 months of free use, but as I mentioned, it's available for the duration of the project. We provide support and training, and then, you know, we will also provide support to help you present the results you generated by using those applications at scientific conferences or at our user meeting. If you'd like to learn more, go to this URL, info.bina.com/research-grant-FAQ, or email me directly, anoop.graywall@bina.roche.com, and I can answer any questions you have about the research grant program or follow up with you after this webinar on the SomaticSeq. Alternatively, if you'd like to just give Bina Rave a try or AIM, go to bina.com/test-drive, and you can start working, running a trial almost immediately. Now again, we're going to start our question and answer session. So just hover over the green bar at the top of your screen to bring out the chat box, the gray box. Click Q&A to answer your, to ask your question, and then choose to send, when you enter your question, choose to send to all panelists, and then we'll go ahead and address it. Again, if you're on a mobile device, the interface is a little bit different. In the upper right corner is where you'll find the option to select Q&A, enter your question, choose to send to all panelists, click send, and then you can actually toggle between your My Q&A panel and your All Q&A panel to see questions that others are asking. All right, so let's get started. Please start asking your questions. Okay, we have a question. What kind of sample did you optimize your classifier to?
Yeah, so the classifier is built based on the training data set. In our own experiment, we used Dream Challenge data sets. We also used in silico titration. So we have tested the classifier based on about 40, 50x whole genome sequencing. There's no reason that it will not work for targeted data sets or our whole exome sequencing. But to do those, we need those kind of training data sets that has the appropriate coverage.
Okay. We have another question. Very nice presentation. When you built the decision tree, how did you determine the order of the nodes? For example, Mutex was the root in your slides. Did you try other combinations, such as VARSCAN2 as the root?
Yeah, so the decision trees are built by the machine learning algorithm. Essentially, it's not exactly deterministic, but the results are almost identical, especially the first few decision trees. It will kind of just pick a variable and then decide based on the data whether it gives you better accuracy. And so Mutex is on top on this decision tree, but in subsequent decision trees there are other tools, other variables. So in this case in particular for this data sets, we really in the ensemble we have a total of 500 decision trees. Each of them has a different weight, and that was the first decision tree. It also was also the simplest one.
All right, another question. Also, your workflow depends on 5 different packages that are maintained by 5 different independent groups. There is difficulty in tracking the updates in different components and maintaining the consistency of your workflow.
Right. So maintaining consistency in the classifier and the test data you apply to is, in a way, very critical. So if you build your classifier based on certain mapping, certain parameters, versions of the tools, then you should also run your test data based on the exact same workflow. So in our Bina array, we have a very good version control system. So you can always run the exact same pipeline for your classifier, for your training data and your test data. So that is a very good point, that the workflow has to remain more or less identical between the training and the test.
Another question, do you need to tune each individual variant caller according to different read depths, et cetera?
So we don't do too much tuning in our experiment. Now, we are aware that if you're doing deep sequencing targeted data sets, then some of the tools need to be tuned to that, and that should be done. The important thing is to maintain the same configuration, same parameters in your training data and your test data. We did not do too much tuning ourselves, simply because our data set is pretty typical, but I know that's not always the case.
Number of mismatches was one of the predicted features, and you only expect one mismatch per read. Do you expect this feature to be useful for long reads with high error rates like PacBio?
Right, so the classifier we presented here is based on short read Illumina data. And then also, for Illumina data, each read has the exact length. Now, so for PacBio, I wouldn't expect this feature to be useful. There will be many more mismatches. Also, it has different, each read sometimes has different read lengths. So basically, that's a totally different kind of data set. And so if we try to apply it to PacBio, we may need an entirely different set of features that's better suited for PacBio.
Great. And another question, will SomaticSeq be available as a tool in the cloud or on premises?
So SomaticSeq will be available in an upcoming release. It should be, when it's released, available in both cloud and on premises.
Next question. How sensitive are the tools to detect different depths of tumor or normal sequencing depths and or insert sizes? Do they have to be the same?
So ultimately the sensitivity depends on the tools that we have incorporated into our workflow. So for read depth that's high, or basically for different data sets, we may have to optimize the parameters for the tools or use different tools for different kind of data. So we are looking into some other tools that's better suited for deep sequencing, low allele frequency data, and then apply the same concept to those data sets. Now, I expect that the feature would be a little different for those kind of data.
Have you looked at Terry Speed's publications building a combined caller? This used validation data from the TCGA endometrial project to determine both false positive and false negative. This tumor type had validation of all the samples for some mutations and all mutations for, I'm not sure if it's complete, but do you understand?
Yes, we are aware of Terry Speed's publication. It is, so we did cite this paper in our own publication. And so the difference is that, so we basically, we use a different machine learning algorithm, and that's actually a very good study. We use Dream Challenge because we know exactly what the ground truth is. There is some issue using TCGA data and using validated mutations. The issue is that those validated mutations came from a call set, and that call set may not be perfectly accurate. For instance, if you use MuTech and VARScan to call your mutations and then validated calls within that call set, then you're only validating what those two callers called, and you don't know the entire ground truth. So your sensitivity and precision calculation are not entirely accurate. The advantage is, of course, it's real data. So yeah, we are aware of the advantage and disadvantages of using TCGA data.
All right, another question. How to avoid overfitting to training set? Which parameter to twist?
How to avoid overfitting to training data. So in machine learning, there is a risk of overfitting. So in our own experience, we split data into two halves, and then one of the parameters in the machine learning algorithm is you can choose how many decision trees you want, in your classifier. And so we chose 500. That's because at 100, when you go from 100 to 100, the results get more accurate, and when it gets to 500, that's about the most accurate it can get. If you try to build more trees to get, more accurate results in your own training data set, if you have 1,000 decision trees, then you actually introduce more noise, and the results does not get better, and sometimes it gets worse. So it is a concern, and you can kind of figure out how much training you want, how many trees you want, and just by looking at the cross-validation data, and make sure that you are not building too many trees based on features that just happen to work in your training.
Okay, another question. How did you select these features? Did you use a feature selection method or pick them by hand?
Yeah, so it's kind of picked by hand. And so some of those feature selections are intuitive, some are by experience, others are really what's in the literature. Strength bias, read depth, and mismatches. Those are kind of well known in the community, and many tools have hard filters built in based on those features. So we use those features. The difference is that we don't apply hard filters, but we apply kind of the probabilistic decision making based on those features.
Okay, another question. Can SomaticSeq be applied for detection of other types of mutations, such as for detection of somatic structural variants?
So we are currently developing ways to detect somatic structural variants. Some of the features that's in SomaticSeq, so far it works for SNVs and indels. Some of the features may be also useful for structural variants. Some of them may not be, so it's currently under development right now.
All right, another question. Is the sample coverage critical to detect the variants using SomaticSeq? What's the minimum coverage for either whole genome sequencing or whole exome sequencing?
So what's the minimum coverage? That kind of depends on how the tools are designed, if the tools can detect mutations at this coverage. So typically, when you look at somatic mutations, 30x is pretty much the minimum you can maybe go. So in the genome biology paper, we also have a study really when we look at the accuracy based on coverage and variant allele frequency. So if you want to detect like high variant allele frequency, like major mutations at 50%, then you can have your coverage at 10. But if you want to detect something like about 10%, then the coverage needs to be higher. So there's no hard number. I would say just a rule of thumb. When people do whole genome sequencing, it's about 30. So if you do whole exome sequencing at 30, you will also detect those mutations. But if you want to detect lower allele frequency, then you need higher coverage.
Another question. Are you able to predict the false negative rate?
False negative rate, so how many mutations you miss. So because in the data that we have presented, we are using Dream Challenge as well as in-situ titration where we know the ground truth. So we could predict false negative rate. So the false negative rate in a way really depends on how many mutations that all callers have missed. And I remember for Dream Challenge stage 3, out of the whole genome, there were about maybe 100 mutations missed. So that's 100, and so that's why we don't have 100% sensitivity. it's 90-something%.
Okay, another question. Do you have something like SomaticSeq for germline calls?
So far we haven't. Germline callers are pretty accurate right now, even without combining many callers. But we may do that in the future. I know there are like have-to-have callers, free base, and others that may improve the results.
Okay, another question. How did you determine your truth set for training? In other words, how did you know for certain that each false positive was in fact false and each true positive was true?
So the question is that, so in the data that we have presented, because the ground truth is built in a way synthetically, so we know what mutations are put in to Dream Challenge, then it's assumed everything else, if called, will be a false positive. And likewise, for in silico titration, we have built a gold set of known ground truth somatic mutations. So another way to apply SomaticSeq would be to, in a similar way, if you don't want to use synthetic data, is to really, run the workflow on the calls, run the workflow on the data, get a call set, and then randomly pick, say, 100 or 200 mutation calls, validate them with the orthogonal methods in order to have really ground truth, know for sure what is a false positive and what is a true positive, and then use that as a training set. So SomaticSeq does depend on a training set that is accurate, and so you want to have very good confidence in your training set that true positive really is true positive and false positive really is false positive.
Okay. We have somebody located internationally who may have to miss the rest of the Q&A, and just want to let you know that the recorded webinar will be available later, so you'll be able to catch the rest of that when we make that available to you. What is the lowest variant allele frequency you're able to reliably detect using your current trained parameters? Also, what is the maximum length of the indel you can detect?
So in the data that we have tested, we have about 30-40x, and the lowest that we have tested, lowest allele frequency that we have tested, is about 10%. And so with the amount, with the amount of coverage we have, 10% is an average of 3 reads. And the lowest we have detected is like a single read, but with really good quality reads. So what we could really detect reliably with the coverage that we have is really about 10%. You could always detect lower allele frequency, but you need higher coverage. And so the maximum length of indel is so tricky. So for deletions, it's indel, a large part still depends on alignment. So for deletion, Vardic, for instance, can detect up to like 100 base pair deletions. Insertions is a little less. You can occasionally detect insertions of 50 base pair or longer, but most indel detected are less than 20 base pairs.
Okay, our last question for the session. It's not clear to me how to obtain a training set for different data sets. Can you elaborate on how to create a non-dream training data set?
Yeah, so training set is probably the most important thing in this workflow. I can think there are 2 ways to do it. So Dream Challenge simulated their mutation using a tool called BAM Surgeon. Basically, it takes real data and spikes mutation in. So the advantage that because you're not synthesizing reads entirely computationally, but you're taking real reads and put mutation in, So it captures a lot of the false positive. It captures a lot of sequencing artifacts that you will expect in the real sequencing data. So that is a way. Another way, which I think is better, is that if you are doing a lot of sequencing, if you're doing, have a huge sample size, the best way to build a classifier based on your own data is to randomly sample 100 to 200 mutation calls from your own data sets. And this will ensure that your training set has the identical characteristic to the rest of your data sets. So you randomly take some mutation calls, validate them, and so you know that for those data sets, what is a true positive and what is a false positive. Then you can use SomaticSeq to build a classifier based on that training set, and then you can apply it to the rest of your data set. And that really is the best way to apply SomaticSeq to get the most accurate result, because in this case, your training data and your test data are identical.
Great, thanks, Lee-Tai. We're going to wrap up now. There are a few folks with some questions we weren't able to answer. We'll follow up with you directly for that. I just want to remind everyone you can get the article available via open access from Genome Biology, so just Google search SomaticSeq or search on PubMed. You can test drive Bina Rave and just go to our website to do that. And consider applying for our Bina Research Grant Program if you have a next-generation sequencing project where you could use a number of our tools. Thank you again.